Okay, Maria Lupiola from Argentina is simultaneously a professor at the Catholic University, is that? Yes, and a member of the staff and has a new job at Monsanto, a second job at Monsanto, where she's creating a new sustainability program that isn't even available in American Monsanto at this time. She was involved in the documenting the initial gene flow of a transgene, the first gene flow of a regulated gene into natural populations. So she's going to be telling us about it.
Thank you, Norman. Thank you for being here, joining us in this great symposium. Today, as Norm said, I am going to be sharing with you my findings during my PhD at Oregon State, where I was working with gene flow from transgenic creeping bentgrass into compatible relatives. So I think one thing that it had been mentioned in talks throughout this symposium today is that, well, gene flow occurs, but then I think it's important to point out that in general we look at or we consider, we think about gene flow through pollen, right? Through pollen between plants of the same species or between plants of different species, right? But it's important that we consider the other means of gene flow, mainly not only in distance but also in time, that is gene flow through seeds or vegetative propagules, especially in perennial species. So that would also result in the dispersion and the changes in that gene frequency in the populations. And I will be giving you some examples of both or different type of means of gene flow in the agroceries complex. So to put this into context, for those of you that don't know creeping bentgrass, creeping bentgrass is Agroceris stolonifera. It's an outcrossing wind-pollinated species. It's allotetraploid, so it's quite diverse. It's perennial, and it does have stolons. It has very small seeds. You can see here, you can have 13,500 seeds per gram of seeds. So seeds are very easily dispersed by winds. It persists outside population field, production fields, so it can get established and persist in the environment because it is a perennial. And it also produces hybrids with other agroceries and polypogon species. In fact, creeping bentgrass or Agroceris stolonifera is part of a Pengrass complex. There are at least 12 of these species that are known to exist in the US. Here is a simplification, and there is not exactly, the origin of each of those species is not exactly known. We know that there are different genomes involved and they are combined. Most of them are polyploid species, so again, quite diverse. And today I will be focusing on these three ones here: creeping bentgrass, redtop, which is Agroceris gigantea, and then rabbit foot, that is Polypogon monspeliensis, right? A species from a different genus. So my study was based on a unique opportunity, I would say. In 2002, 4,500 hectares seed production control area was established in Oregon. This is a highland desert irrigated area where agriculture it takes part. And for those that are not familiarized, this star here in the state of Oregon is representing the location of the area. And within that control area, 162 hectares were planted to transgenic glyphosate-resistant creeping bentgrass in 2002. These 162 hectares were separated in 9 fields that are here exemplified by the squares. The whole area had very strictly production practices put in place, and also a mitigation program. And the seeds were produced for only those fields were allowed to flower, produce pollen and seeds for only 1 year, and then in 2003, after harvest, they were taken out of production. Right? So we can say that this was a unique opportunity to study the gene flow of this transgene and its integration at the landscape level. So today I will describe 4 of the objectives and the approaches that we took to answering each of these objectives. The first one was determine if gene flow really was occurring in there, so determine the proportion of glyphosate-resistant plants, transgenic plants established outside of cultivation. For this, we conducted a 4-year in situ survey. Then, to assess the occurrence of pollen-mediated gene flow, and for that we did greenhouse screenings. Then, to confirm the putative intergeneric and interspecific hybrids, and for that, because the great diversity in this complex, we needed to develop molecular markers to be able to identify species and then hybrids between these species. And we developed both nuclear and chloroplast markers, SSR markers. And then finally study the potential of seed-mediated gene flow. And for that, we look at how the seeds will behave under at being soaked at water and for how long they can germinate and how they can spread around. So for the first objective, the in situ survey, we surveyed the area between 2003 and 2006 during summer. We walked all the ditches and irrigation canals in the area, looking for plants, and wherever we found a creeping Pengrass plant or a relative species, we will check with the trait checks, grab some piece of leaves and see if you have one line, it was non-transgenic and susceptible to glyphosate, and if we had 2 lines, that was a confirmation that that plant was transgenic, so it was carrying the transgene. Each plant was georeferenced, and then we collected panicles and green tissue for those plants that we were finding. So this is a summary of the results, and here you will see the 4 years, and the first column will be the susceptible plants, like non-transgenic plants that we found, and the R would be the glyphosate-resistant plants that we found. And then you will see 4 different colors. The green ones are creeping Pengrass plants that we tested, the pale blue red top, the purple rabbit foot, and the gray ones were agrostis. In that case, we weren't able to identify the plant to a species. We knew that it was agrostis genus, but we weren't able to get to the species by the identification of the plant at that moment. And so the bars will show the plant's number for each of these different types. So in the first year, as we were expecting, we only tested 57 plants, most of them red tops, and all of them were non-transgenic, right? Susceptible. In 2004, we tested 300 plants, and this is again in irrigation ditches and canals. We didn't go into production fields, just stay on the margins. Out of 300 plants that we tested, 46% were resistant, glyphosate resistant or transgenic. And out of the total of creeping Pengrass plants that we tested, 93% of them were transgenic. So that meaning that, well, yeah, the transgenic plants were out there, right? In 2005, we intensified the survey quite a bit, and we tested 1,290 plants in situ, right? And out of that total, 40% were glyphosate resistant, and again, within the creeping Pengrass, 54% of the plants that we tested were transgenic or glyphosate resistant. And then finally, in 2006, we tested 1,073 plants. 34% were transgenic, and then out of the creeping Pengrass, 62% were glyphosate resistant. This is just to show how the canals look like, the ones that were easily to see the plants. Some were larger, some were really tiny. So everybody was counted. And then we also saw many of the plants that were producing seed panicles and seed at that time. So this is a map, again showing the control area with the fields that were in production in 2002. Remember, these were taken out of production in 2003. This is the map of 2006, and the red triangles show the location of the transgenic creeping Pengrass plants, and the yellow circles show the location of the acceptable plants. The further plant that we found was this one here, that was out of the control area and was 4.5 kilometers away from the border. We don't know where it came from, but that was the farthest one that we found. We didn't walk further than that either. And this is more or less the same time Watrud et al. Were doing a gene flow study, but focusing mainly outside of the control area. And what they found, they put some sentinels plants that they set up in there, and they found pollen movement up to 25 kilometers going to a process stoloniferous plants, and then pollen movement up to 14 kilometers on resident red dot plants that were in place. So one first thing to know is that we will never know how far the last pollen grain. So moving on to the screenings result in the greenhouse, we tested a lot of seedlings coming from different mother plants from different species. And I'm not going to go into detail for each of the species, but if we look at the total, we can say that in 2003, out of nearly 16,000 seedlings screened, only 58 were transgenic. So that is 0.36%. In 2004, out of 207,000 seedlings, 66 were resistant. Out of 44,000 in 2005, 16, and out of nearly 105,000 seedlings in 2006, only 6 were transgenic. So if we compare the 0.006 that will be gene flow through pollen to the 63% of transgenic plants that I was finding in situ, we can see that there is a huge gap. So what's happening in the middle, right? The take-home message here is that yes, gene flow pollen was found all of 4 years, and the percentages were more or less similar to what you find in literature about gene flow through pollen. So then another objective that we had was like, okay, we were seeing some seedlings that were transgenic produced in other species. So we wanted to confirm those interspecific and even intergeneric hybrids with molecular markers. So we had 35 plants that we were thinking that were crosses between red top and creeping Pengrass that were transgenic, and these were again plants resulting on panicles of red top plants that were collected in situ, that were yielding transgenic seedlings, right? So we look at the chloroplast SSRs that we developed, and also we identified a MADK deletion that was very useful, and those tools together were able to separate the red top from the creeping Pengrass chloroplast, or cytoplasm. And then we look at nuclear markers and also sequence the ITS region. And for that, in this case, we were able to find both sequences of red top and creeping Pengrass in the nucleus. Here we didn't sequence because for each plant we were sequencing at least 10 clones, and was kind of a quite intensive process. And then in this case we find all creeping Pengrass ITS. And then the ideal marker that we had was the transgene, right? And we also ran PCR to confirm the presence of the CP4 in the SPS gene. So from here we were able to confirm the glyphosate-resistant or transgenic interspecific hybrid between red top and creeping Pengrass, red top being the mother plant and receiving pollen from transgenic creeping Pengrass that were produced in situ. And in here, because we were thinking about a back cross of already hybrid to creeping Pengrass, we weren't able to confirm this because we weren't finding any ITS sequences from red top in that population, and also the nuclear markers were not strong enough to call this out. But these plants have rhizomes, which is a phenotypic characteristic of red top. So still have a question mark in there. Probably we have to do more sequencing there to try to find a red top sequence. So we confirmed 35 glyphosate-resistant red top by creeping Pengrass interspecific hybrid produced in situ. We were not able to confirm back crosses to putative hybrids. Then within the established plants, we confirmed one creeping Pengrass by red top plant that was already established in situ, but was acceptable. But again, because of this same problem, we were not able to confirm other 5 putative hybrids, 4 of which are transgenic. But again, phenotypically, they look like something has been mixed in there, but molecularly, because of the great variation, we don't have the power to call them apart. But another interesting thing that we found, and this was completely random, because it was produced on, it was a panicle, seedlings that came out from a panicle of a transgenic creeping Pengrass plant. And when doing the counting, I found kind of a weird phenotype, so I kept it. And then I left it there in the greenhouse, and all of a sudden, it produced a panicle. I'm like, what? Creeping Pengrass needs vernalization to flower, and that plant was not vernalized. But it did, indeed, it produced a panicle. And that panicle was really weird looking. This is a typical creeping Pengrass panicle anthesis. This is rabbit foot, Polypogamon speliensis, and this was the panicle that I saw. And I was like, what is this? So I tested the flag leaf with the trait checks, and it was resistant. So then I went over and looked at molecular markers for it, because we were seeing some mixed phenotype there. It is perennial, it has stolons, which Polypogamon speliensis doesn't have, and it has on blooms, which creeping Pengrass doesn't have. So looking at the markers, we found that for creeping Pengrass, we are expecting this type of chloroplast SSR profile and the MATK deletion. For red top, we have a really clear different chloroplast profile, and also the MATK shorter segment, and this was what we were finding in the putative hybrid. Then for the ITS sequence, we found again a combination of both creeping Pengrass and rabbit foot sequence in the nuclear ITS sequences. So, and also we were having the nuclear marker for the transgene. So indeed, we were able to confirm that this plant was an intergeneric creeping Pengrass by rabbit food hybrid that was produced in situ. And so molecular markers and also ITS confirm that there is great diversity and promiscuity within the Pengrass complex. And so our final objective was to see, okay, can we explain that difference between the percentage of gene flow through pollen and what we were finding in situ? So we look at how much the panicles can move in the waterways that are characteristic of that area, and we threw some panicles in the ditches, and we follow them, and we got an average of 18 meters per minute of movement of the panicles in the waterways. So then we went like, okay, so for how long can the seeds survive after being submerged in water? So to give us an idea of how far they can get. So we did this study using non-transgenic creeping Pengrass cultivars, 3 different cultivars, and we put the panicles in water, in containers in water at 4 degrees Celsius and 20 degrees Celsius, right? And the first week, second week, 4, 6, 8, 12, and 17 weeks after putting those panicles in those containers without changing the water, we would take out seeds or panicles, crush them, dry the seed, and conduct traditional germination tests. And so what we saw was that for the panicles that were kept in water at 20 degrees, germination stayed up to 95%, even after 17 weeks of having the panicle into the water. I can tell you that was a gooey mass of things, because you couldn't tell the panicles any longer. We would look like that and dry those seeds and then use them for germination tests. The interesting thing is that the ones that were at 4 degrees dropped their germination down to 50%, right? So the question was, okay, so are these seeds dead, or can they still germinate? So what we did was we took these seeds out of the water and we stored them for 9 months in dry paper bags, right? And after those 9 months of dry storage, we performed the same germination test. And what we found is that here we also have control seeds that weren't submerged in water at all. That is this first line. Then the ones that were in water for 17 weeks at 20 degrees, and the ones that were in water for 17 weeks at 4 degrees. And we found that the germination was up to 87% again. So the seeds were not dead, they went into secondary dormancy. So that is another interesting thing to consider. So this confirms the great potential for gene flow through seed movement, right? And those seeds can be moved either by wind, by water, by animals. But we do have to consider seeds as a means of gene flow in time and space, and even continents, if we think about seed movement and shippings. So, and this is an example of selection pressure, what selection pressure can do when you don't spray glyphosate on one side of the ditch and you do spray glyphosate and have the transgenic glyphosate-resistant species in there, or population in there. This is a picture of summer of 2010. So general conclusions, the gene moved, and even after 3 years of mitigation, 62% of the plants found out there were transgenic. Pollen-mediated gene flow occurred all 4 years, but was not enough for explaining all the plants that we were looking or finding at. The non-glyphosate interspecific hybrid established in situ were confirmed. No resistant transgenic interspecific hybrids established were confirmed, but we confirmed several glyphosate-resistant interspecific and intergeneric hybrids that were produced in situ. And then the sow seeds maintain their germination potential, therefore gene flow through seed must be regarded as an important means of gene flow. And this is another interesting thing. Here we were able to do all this because we were tracking a transgene. So transgenes definitely facilitates gene flow studies, but gene flow, as we saw earlier, is not exclusive for transgenes, right? So what am I using this knowledge now to? So considering that gene flow occurs both in space and time, that the species characteristics, mainly their reproduction system, the compatibility with others, their cycle, their seed size and variability, impact potential gene flow, and then that is key to identify those dispersal mechanisms and available niches for those species or seeds or propagules to establish, that wind, water, and animals play a role in facilitating gene flow, and in general we cannot control those factors. The fate and the impact of the gene flow on the environment are related to the trait that that gene encodes and also the selection pressure that we put on it. And so considering all these, we can say that gene flows matters, right? And it matters in particular from what I am doing now from the agronomical perspective. And so we're using that information to now designing systemic approaches for weed management, using this information to reduce the chances of selecting and also dispersing herbicide resistance in weeds. And for that, I want to thank you all for being here and sharing this time with us, and I hope I have some time for questions.
Yes, we have time for about 2 questions. Are there any questions for Professor Lupiola?
Really great talk. I'm wondering what those mitigation practices that seem to be so ineffective at controlling the spread of the transgene were.
Well, the main one was to remove all the transgenic plants that were found. And in fact, when I was doing this work, we were facilitating all the data points for the company that was doing the mitigation at that time. And in many cases, we went back to the locations of where we had found plants the previous year, and you found, well, in some cases there weren't any plants any longer, but in some other cases, the plant was still there. And I think one key element that has to do with that is that it is a perennial. So one stolon, one single node, one single tiller that stays there and you don't kill, it's enough for allowing that plant to go over time and stay established. And so that was the main mitigation practice, was just to remove plants. And they were doing that by different a different set of approaches. One was using alternative herbicides that in some cases have very good effect. The problem there is that you have to get to the plant at the moment that is quite susceptible to that herbicide, and if you get it later in the season, it's too late. They were also burning the plants earlier in the season to prevent flowering, and for example, those really big plants that were 1 meter wide, I don't know if it was one plant or many, but those are not easy to kill with alternative herbicides. So they will go and burn those down to at least prevent the seed germination and the pollen production that year. And the other way was just digging them out and taking them out of the area. In some cases, growers were also taking them out of their ditches, But we also found that if you take it out and just flip it over and leave it there, they will re-establish. So you will not kill them that way. So yeah, it's a very complex, systematic, if you want, approach to try to combine different methods to try to keep it low.